Okay, good afternoon, everybody, and I hope that you're all well. Onto our second class today. We have a lot to cover today, but just before we start, there's a couple of housekeeping things that I want to discuss with you. Please make sure that you have read through the syllabus, which is posted to Canvas. But I have I left in a section there which actually applies to other classes, which I need to take out, which I just want to bring your attention to. And this is the examination makeup policy. That whole paragraph comes out because in the accelerated sessions, there are no makeup exams. No makeup exams, none. Remember, please, to read through the way in which grades are going to be computed. And 60% of your final grade comes from the tests which you do each weekend. So you'll be doing 4 tests. One of those tests, the lowest score, will be dropped, leaving 3 tests of 20% each, okay? So if you miss an exam, that is your dropped exam. No matter how low any of the other exams are, don't do it, okay? Do not deliberately miss an exam. You're not allowed to do that. But I realize that of the 4 exams, the lowest score will be dropped. 15% of your grade is a cumulative exam, which is administered at the same time as the very last test on your final exam day. I'll explain later on exactly how that works. And then I want to bring your attention, please, to the fact that you need already to be going onto YouTube or wherever to find a particular true crime that you are going to report on in your term paper. That term paper needs to be sent to me. I'll post the final date, but it's about 5 days before the end of class. It's that weekend before the classes end is when it has to be submitted to me. To do that, you need to, you can do however you like. You can take a TV episode, not fictional. It has to be documentary or a true crime book, whatever, and briefly present the circumstances, etc., but concentrate on the evidence that is collected, on how it was collected, and on its significance in the case. Okay, I don't just want an explanation of the circumstances of the crime. I need to, we are engaged in a study of criminalistics. I want an in-depth explanation of how evidence was collected and what its relevance is to the particular crime. I would like you, please, to let me know just in a paragraph, emailed to me by July 11th. Make a note of the date, July 11th. You must please send me a paragraph which explains which crime you are going to be investigating. I strongly advise against choosing a very complex crime like a serial killer or something like that, because it's just too complex for you to explain within the bounds of the time and everything that you have. Please just read carefully on your syllabus it tells you what to do, and that is 3 to 4 pages, and you will automatically gain points if you use more than one source for your information. All right. Okay, so that aside, you can always just email me if you're confused. All right, we're going to carry on with our discussion about the history of forensic science. And this is for an important reason, is because at each point here, I've introduced the advent of techniques which are of particular significance even in modern day investigation, and how they were arrived at. But within most of our lifetimes, there has been a huge revolution in forensic science because of the advent of DNA analysis, and in particular, our ability to sequence DNA. Now, let's just think for a minute about what DNA is and what its significance is, but we will discuss this in depth in another series of classes later on. So DNA is the hereditary material. It is a double-stranded molecule. There's one side here, and here's the other side here. And it is the hereditary material because it essentially contains the recipes in code for all of the functioning of our cells. It contains, in fact, the recipes for all the proteins that we use to drive our metabolism, growth, and everything else. So the structure was elucidated by 4 workers, not 2, but by 4. Watson and Crick are the most famous of the workers, but the other 2 were Wilkins and Franklin, who seldom receive the credit that they are due. Watson and Crick always pull the limelight. They were very good at publicity, and it paid off for them. So they were the 4 of them actually arrived at an explanation of the functioning of the structure and functioning of DNA as the hereditary material. The important thing about DNA is that if you look across here, you see these across here. These are structures which are very rich in nitrogen. And there are 4 different kinds in DNA. And they can occur in any order, but it's their order that specifies the DNA code. And because of what happens during sexual reproduction, each individual ends up being a mixture of DNA from mother and father. And the DNA sequence, the sequence of those bases in every individual is completely unique unless you have an identical sibling. If you have an identical twin or identical triplet or whatever, then they share the same DNA sequence. But otherwise, it can be computed what the chances are of you having an identical sequence to another human being. And this is 1 in hundreds, thousands of trillions. So the chances of you having a matching DNA sequence in somebody else somewhere else is vanishingly small. So we can truly we can proceed as if each individual's DNA sequence is absolutely unique. Now, this was a hugely important finding because DNA is very frequently shed at a crime scene. Remember Locard's principle, okay? Every criminal leaves something behind and takes something away. Well, what they may leave behind may have DNA in it. At its very simplest, let me explain to you, for example, as human beings, we shed skin flakes the whole time. Those skin flakes contain DNA. Now, unfortunately, our techniques are not good enough to use skin flakes as a source of DNA, but it's just an example to you that, in fact, we shed DNA inadvertently the whole time. But in commission of a crime, very often liquids such as blood, semen, other body saliva, other body fluids, which are actually rich in DNA, may be left behind or may be taken away from the crime scene by the perpetrator. And using DNA, we can link suspect and crime, but if the DNA matches. So the really important thing about DNA is that it provides evidence with a very, very high degree of accuracy and a very low probability of error. And we can compute that. We can actually present to the court mathematical bounds and say the likelihood that this DNA matches another person who could have been the perpetrator is 1 in 23 billion or whatever. That is hugely important. This is truly scientific evidence. The fact that we can say what is probability of error is very, very important. Now, initially, as you will see, in the first crimes that were solved with using DNA was in the 1980s. And at that time, you actually needed quite a lot of DNA. A semen stain, a blood stain, these sorts of things would provide enough DNA for it to be analyzed. But later on, techniques have been developed where extremely small amounts, so-called touch DNA, can be extracted from a scene. So, for example, if somebody has gripped a gun butt in firing it and then has left the gun, we can actually very often, not always, but very often obtain sufficient DNA from the gun butt to actually identify who it was that held it. So DNA has become enormously important. But it has to be said, DNA is not left at every crime scene. Despite the fact that we shed skin flakes, it's no good to us because how do we find the skin flakes? And if we found the skin flakes, there's not enough DNA on them to use. But the sufficient DNA is not always left at a crime scene. In fact, many, many, many crime scenes, there is no DNA trace left or taken. So it's only useful where DNA has been left behind and where we can actually match that DNA. So this is the scientist who was the very first to use a DNA, it was a DNA fingerprinting, they called it. It's a DNA profiling system that he devised. And this points to the real importance of all evidence, because this is Professor Alec Jeffreys, and in Britain, he devised this to investigate a particular case where 2 girls had been murdered, and it was assumed that they were murdered by the same person. And he devised this technique, applied it to evidence from the scene, and was able to exonerate a person who had been accused. He was actually able to exonerate somebody who the police were very, very firmly convinced was the perpetrator. Ultimately, in addition, it led to the conviction of the correct murderer. And this was his techniques were used for a long time. They're no longer commonly used because they needed such a lot of DNA to perform the test. But these were the very first DNA cases solved in the laboratory. This is the guilty party. You can read this, but we'll actually see a movie later on when we talk about DNA. We'll see a movie about this case. This is Colin Pitchfork, who was the murderer of these 2 girls. Okay, so you'll begin to realize that as forensic science has developed, sub areas of investigation have developed which require considerable expertise. And it passes beyond the scope of, say, a police precinct or a police investigator to be able to perform these kinds of tests and these kinds of functions. So instead, increasingly, there have been established on a state or federal level laboratories where experts can gather, where this evidence can be processed. And the first ones in the United States were were established as on a federal level in 1932 by J. Edgar Hoover. And the FBI laboratories, the Federal Bureau of Investigation Laboratories, are still very, very active and very important, because the whole field looks to the FBI to develop techniques and to establish the standards for techniques, in addition to the fact that they themselves, of course, are doing investigations the whole time. I'm not going to play it now, but please go to this link here. It's a very short movie just about some of the things that such a laboratory does, what the FBI laboratories actually cover. So these laboratories have become increasingly important. First of all, because courts began to address the issue of the scientific evidence which was being presented to them. Because there is this problem. Just because somebody stands up in a court of law and says, I am an expert in this field and this is what I have found, or this is what I think, should not just make it so. There should be standards around which we judge scientific evidence very carefully. And there should be standards that the courts can refer to as to whether they admit evidence or not. So the Supreme Court, there have been various decisions that you'll hear about, but in the 1960s, particularly the Supreme Court began to address this and increasingly emphasized the importance of methods and evidence being scientifically evaluated. In other words, this should not be the evidence, the methods, whatever, should not just be arrived at by an individual in isolation. Instead, it should be a matter of referral to the wide scientific community. We're going to hear in a little while but what we actually mean by science. Because in forensic science, unfortunately, there are areas where true scientific principle has not been applied. And this has sometimes led to disastrous results. But the courts have increasingly addressed this issue and tried to say what it is that makes something scientific and what scientific evidence is actually admissible in the court and who should do that admitting. Now, this is becoming increasingly important because there had been for a very long time increasing crime levels, particularly from the 1960s to the end of the century. First of all, because of the increase in drug usage and in drug trafficking, illegal drug trafficking. That was the first thing, which fed an entire crime wave. The second thing is, particularly in the United States, the widespread availability of guns and their usage in commission of crime, which is quite exceptional actually in the world in general. All of this burdened investigative services, absolutely burdened ordinary precincts. What a precinct would have to do would be, under old circumstances, we would have investigators investigating a crime. They would acquire evidence which they themselves were not able necessarily to analyze. They would have to find somebody who would do that analysis for them, then send them the results, and they would then go and put that together, and if they could arrive at a suspect, present it in court. Well, that's a very inefficient system if all of the burden falls upon an investigating officer to acquire all that information, synthesize it, and present it. This became even worse once DNA profiling began, because DNA profiling is a tedious and lengthy process which needs to be conducted under very strictly controlled conditions. So instead, increasingly, on a statewide basis or sometimes on a municipal basis, crime labs have been established where experts can gather who perform all of those functions and efficiently refer their findings out to investigating personnel. And that is the basic function of a crime lab. It is a gathering place for experts in all of these fields who can process information, process the evidence. And so again, here's a little short link for you to just have a look at. And again, here are some of the things that occur in a crime lab. The oldest crime lab is actually in Los Angeles, established in 1923. And establishment of crime labs has proved to be a very important feature of criminalistics because of the fact that they tend to centralize a source of information. Throughout the United States, crime investigation and policing is extremely fragmented. It's fragmented from the basis of individual precincts, operate as independent organizations. And municipalities may have some level of organization, but they operate independent of other municipalities. States operate independent of other states. And there is actually limited federal coordination of these investigations, except under special conditions. So the crime labs begin to perform a really important function of centralizing information. You may have 20 precincts in a city and one crime lab to which all of the evidence is sent. That serves a very useful purpose because it means that through that crime lab, the precincts may begin to talk to one another about evidence that they are accumulating. There are 411, I think this slide was written some time ago. There's probably a lot more by now. Let's say 500, around 500, I would guess, public crime labs, which operating at all sorts of levels throughout the nation, at federal, state, county, and municipal level. Unfortunately, it has to be said that sometimes there are breakdowns, significant breakdowns in communication between those labs even. So that itself is a problem. Oh, sorry. So yes, this is a repeat slide, but it's got this important link here. I asked you yesterday to watch this movie about this particular case. And that was for two reasons. First of all, that movie introduces you to several lines of criminalistic investigation. Fingerprinting, blood work, guns, etc., etc., etc. So it just shows you for a crime how complex the gathering of forensic evidence actually is. But there is another reason why I wanted you to watch that. And that is because embedded in that investigation is an example of how a crime scene can be investigated in a scientific manner and quite apart from everything else. So when you think about that particular crime scene, they gathered evidence, and then what happens to that evidence? Does the investigating officer sit and do the investigating? No. The investigating officer refers the evidence that they have gathered to various units. This can be within their precincts. Some of these may be within the precinct, but as I've said, increasingly that it would be referred to a lab, to a crime lab where somebody is dedicated to examining a particular line of evidence. These are, your book says there are 5. I say that there are 6 basic services, which any precinct would make use of. The first is a biological unit. And these are people who are involved with in looking at any product of a living thing. So they are looking at body fluids, hair. They may also look at things like fibers, although some units have a dedicated fiber unit. These are the people also who will see to it that blood, semen, whatever is subjected to DNA profiling, a DNA analysis. The firearms unit investigates guns and the products of use of a gun in a crime. Discharged bullets, guns themselves, identifying ownership, identifying whether a bullet has been fired through a particular gun, etc., etc. Document units examine questioned documents. A questioned document is any document which is believed to may provide evidence in a crime of any sort. This, now you may immediately think, well, this means fraud and this kind of stuff. Certainly it does, but in a surprising number of cases, documents gathered at a scene or documents accumulated around a particular crime provide very important evidence. And those would be processed by a document unit. Photographic units, now this is something a number of you may want to prick your ears up at. Photographic units have been hugely important in investigations since photography was invented. And the reason for that is that by photography, a documentation can be preserved of the crime scene at its various stages, as it is discovered before anything is moved, as it looks once maybe a body has been removed or whatever, has been partially processed and other evidence has been uncovered. The photographic unit nowadays do two things. They do still photography, of course, which is the standard and classic way of recording the crime scene, but they also do videography. And coupled with the two now very frequently is computer reconstruction of crime scene, which can be extremely useful because it means that in a court, a jury or a judge and a jury can be actually led through a crime scene on screen, which may be extremely useful. Crime scene photographers, there is training available. There are courses available for people to train as crime scene photographers. Some of you may be interested in that as a profession, because many precincts will employ crime scene photographers on their own, quite apart from the fact that crime labs will have a photographic unit as well. Toxicology units examine body fluids, organs, etc., for the presence of drugs and poisons. And this is extremely common. Nowadays in the United States, for example, the commonest cause of accidental death is drug overdose. And that has to be established that has occurred has to be established by the toxicology unit. Lastly, of the six, is the latent fingerprint unit. And the fingerprint is regarded really nowadays as being latent until it is being completely processed and analyzed. So this is the fingerprint unit. And they are present at the crime scene and all the way through the complete analysis of fingerprints. Okay, so I keep mentioning this topic of using scientific method. What is the scientific method? It is a particular set of precepts, that is, of ideas which follow in a logical order. which define what makes something scientific, as opposed to something simply being speculative. An idea is not scientific. A belief is not scientific. The scientific, when you say that something is science, it means that evidence has been gathered using a particular set of constructs. And they are these. First of all, we begin with making observation of some sort. But we then, in performing that observation, we begin to wonder about something, and we begin to formulate a question. And we begin to say, this is something I need to investigate. This is something which is worthy of investigation. And to investigate it, we don't just stare at it. We actually break it into parts. And we begin to formulate reasonable hypotheses, a best guess to answer the question that we have posed. That is what an hypothesis is. It's our best guess at why something is. The important thing in scientific method is that we don't stop there. If you're just ruminating about something, thinking, well, why might this be? Well, this, could be that, could be that. You would stop at hypothesis, but the scientist does not. The scientist tests the hypothesis, and they test the hypothesis through rigorous experimentation or through very, very careful observation if an experiment cannot be performed. And they attempt to judge whether their hypothesis, their best guess, was true or not true. If they accept the hypothesis, it becomes scientific evidence. So I know that this is like woolly thinking, this is philosophical stuff, but let's have a look. Here is the order. We're going to sit and observe a situation. In observing the situation, we are going to formulate a question. It is very, very useful when you formulate that question to then go out and see if anybody else has asked that question. Has anybody else researched around that particular question? If they have, there's your answers may lie right there. Your answers may lie in reading, not in, but very often nobody has researched that exact question that you came up with. So you draw up a series of hypotheses, and the hypotheses will attempt to answer the question. They will provide an answer to the question, but the hypothesis must be testable. And you test the hypothesis with experimentation. Once you've done your experiment, you have to analyze the data, of course, to know what the results were. And you draw a conclusion. And in drawing the conclusion, you either accept or reject the hypothesis that you made here. If you reject the hypothesis, you begin the process again. You push the question through another hypothesis and test again. So, for example, here's a very simple example. You have plants, and you plant them, and you want to know. You understand that there are many different fertilizers at the garden store, which could help your plant to grow. But you have no idea which one is going to work best. That's what is your observation? Your observation is plants grow, and plants grow, or supposedly grow better with fertilizers. But your question, which fertilizer? That's your question. Which type of fertilizer? You hypothesize. I have three possible fertilizers. I hypothesize that fertilizer A will work better than the others. Well, how am I going to test that hypothesis? It's relatively easy. I plant three identical plants, and I feed one with fertilizer A, one with fertilizer B, and one with fertilizer C, and I see which one grows best. In this case, A grew best. That is your result. You look at this result and you say, do I accept or reject my hypothesis? I hypothesize that A would work best. Yes, indeed. The hypothesis is proven correct, and you can draw a conclusion. Use fertilizer A. It works best for these particular plants. What about if we had said, no, C works best? That's our hypothesis. It's just a guess. It could be right, it could be wrong. We test and we find C didn't work well at all. We reject our hypothesis. We return to our experiment and re-examine our results, and we find that A is best. You still arrive at the useful outcome. Sometimes if you reject a hypothesis, it requires reformulating your experiment, rethinking your ideas. But you have, just in this simple example here, you have performed a scientific experiment and you have arrived at a valid conclusion based upon the fact that you drew up a reasonable hypothesis and you accepted it. That makes it science. Not science would be staring at the shelf in the garden store and saying, okay, I don't care. I'm just going to use this fertilizer. Take it home, use that fertilizer only. Your plants grow, and you say, well, that's the best fertilizer to use. Would that be a valid scientific conclusion? No, because you have not performed an experiment. You haven't formulated a valid hypothesis, and you have not tested the hypothesis. So that just making a conclusion with no evidence is not scientific, but we do it all the time. Human beings do that the whole time. We make the mistake of drawing a conclusion even when evidence is faulty, and it's for all sorts of reasons. But in crime scene investigation, it can become tremendously important, because if people draw leap to a conclusion when the evidence is not sufficient to support that conclusion in a scientific manner, they may make disastrously wrong decisions about how the crime investigation should go. And that happens very, very frequently, unfortunately. So just think about that little movie in the back and work out how does that provide you with some scientific principle. Well, first of all, what was the observation? A crime was committed. That's an observation. The crime was committed with a gun. It was committed with a gun. That gun is very important. That gun is really important. What is our question? Where's the gun? Where is the gun? In trying to find that gun, they fortunately were imaginative and thought of the different ways in which that gun could have been disposed of, hidden, taken away, etc., etc. But they formed a very reasonable hypothesis. Around that area, there were many rivers, and the perpetrator had to cross a river to get to the crime scene and cross a river to leave the crime scene. To cross the river, they have to go across a bridge. And the hypothesis is the perpetrator threw evidence off a bridge into a river. That's the hypothesis, a very good hypothesis. It could have been entirely untrue. The perpetrators could have driven off across a bridge and gone and buried evidence somewhere. But the hypothesis is a good guess, and it is a good guess which can be tested by experiment. So what is the experiment? The experiment is to go to a bridge and look in the water underneath. So there's however many bridges. Each one constitutes an experiment. So we're going to say, okay, hypothesis one, threw it off bridge one. Go to bridge one, look in the water. No, it's not there. Okay, reject that hypothesis. Hypothesis two, threw it off bridge two. experiment. Go bridge 2, gather your evidence. There it is. There it is in the water. Accept that hypothesis, perpetrator threw it off bridge 2. And now you've got the evidence which can be processed and then presented in court. The evidence has been gathered because the investigator took time to formulate a truly scientific approach, even though it becomes instinctive. Even though it's instinctive, it still satisfies all of the principles of scientific investigation. But unfortunately, in many forensic investigations, these steps are not followed. And in fact, in some branches of forensic science, true scientific principle has not been followed. I'll give you one startling example. And that is, we always say that no two people have the same fingerprints. That is what's stated almost as a fact, right? But there is no scientific evidence to back that up. There is extensive observation, and the observation has so far, by and large, supported the idea that no two people have the same fingerprints. But let me tell you that later on we're going to hear about a case where, in fact, two people, thousands and thousands of miles apart from one another, had sufficiently similar fingerprints that forensic investigators, numerous forensic investigators, identified the two of them as being the same person when they were not, with very serious consequences for one of them. And here is an example of how we need always to be aware of the fact that we are limited by our technique. Fingerprints are analyzed in a particular way, following particular steps. And it now transpires that the number of steps that we take to analyze fingerprints may not be sufficient to distinguish very similar fingerprints, even though this is one case in billions. We still want to not make those kind of mistakes. So if you proceed with your interest in forensic science and in criminalistics, you're going to have to adopt a branch of forensic science in order to study that further. And you are going to learn that you have to apply scientific principles to all of your analysis, or whatever line of expertise you have. In modern times, this evidence must be presented in court by expert testimony. And an expert, at least one expert, must come to the court and present that. In the old days, they could present a written report to the court through the investigating officers or whoever. That is no longer accepted. That was struck down by the Supreme Court because every defendant has the right to see an accuser in court and defend themselves by argument. But an expert witness is somebody whom the court determines possesses knowledge relevant to the trial, which an ordinary person wouldn't be able to provide by simple observation. Usually, the person will present to the court a brief summary of their qualifications, et cetera, et cetera, et cetera. Throughout this discussion, there's something to realize, and that is that whether evidence can be accepted by the court or not is the role of the judge. The judge decides whether a particular line of evidence can be presented in a case. But the importance of that evidence is determined by the jury. The jury is sitting listening to the expert. The judge says, yes, you can testify. You can present this evidence to the court. The jury listens to that evidence, and they're the ones who have to decide how important it is in determining guilt or innocence. So the expert witness is providing to the court knowledge which the ordinary person and which members of the court are not likely to have. But it's very important in presenting that evidence that to realize that the expert may also be called on to give an opinion as to the significance of what they have found. And in doing so, if it is truly scientific and is presented in a truly scientific way, then the expert will tell the court what the likelihood is that they are in fact wrong. It may sound like a strange thing to have to say, but it's hugely important. How certain are we that what we have decided is in fact true? So the standards for presentation of evidence have been set over a very long time. There are two particularly important findings whose names you should remember. The first is the Frye standard. This is a case where the court set the rules as to what kind of evidence can be presented to the court as scientific evidence. And there had been a series of cases all over the place where people claimed to be presenting scientific evidence, which was not in fact scientific at all. But in this particular finding, the District Court of DC, Washington, DC, established that the admissibility of scientific evidence depended upon the opinion of the scientific community. In other words, the evidence must be generally accepted by the scientific community. That, in fact, is a very, very strict finding. That means, in fact, that this places a stricture on development of new techniques. It means that new techniques in forensic science must be extensively investigated, extensively published in the scientific literature, extensively accepted before they can be presented in court. So it was regarded as being very constraining on the kinds of evidence that could be presented. But much later on, in 1990s, in this case, Daubert versus Merrill Dow, the Supreme Court actually found that the Frye standard, it had been set by a district court, not by a federal court. Although courts all over the country began to use the Frye standard. In this case, the Daubert finding was that it's not an absolute prerequisite that the whole scientific community accept. Instead, the onus falls on the trial judge to decide whether or not scientific evidence is acceptable. So it opened up the field a lot, because this meant that a trial judge could allow a new technique, evidence gathered through a new technique, to be presented in the court, and the trial judge could listen to the argument and decide whether or not that should be admitted into evidence. If the trial judge decides not, then she can just say, jury disregard that. But it places the onus now falls on the trial judge to be the gatekeeper of deciding what constitutes scientific evidence or not. But it's not just left up to the judge off the top of their head to have to decide this. There were standards recommended by the Daubert finding. And was that the judge should base their decision on the following: whether or not this technique, even if it's new, has this been properly scientifically tested? Second of all, has this been peer reviewed? In other words, have people in the field at least looked at this and accepted it and the way in which it was developed? Even if it's not extensive, has it been peer reviewed? And especially if it's been published, and then it acquires a cachet, which makes it more likely to be accepted. Very, very, very importantly, this is probably the most important thing of all. The judge is called on to decide whether rates of error have been established. How likely is it that a finding could be wrong? And is it very, very likely that it could be wrong? It's probably not a good idea to accept it. If it could happen that it's wrong, but it's unlikely, then that can be told to the jury, right? We are relatively sure within these bounds that this is correct. If it is wrong, it is the error rate is small. It's really important to know under what conditions should these tests or whatever it is be conducted, because once the court has accepted a particular line of scientific inquiry, that establishes a precedent for other courts to accept similar evidence. But it's very important that the investigation be conducted under exactly the same conditions in the laboratory. The laboratory examination be conducted under the same conditions every time. So there needs to be a quality control established, even if different laboratories are doing a particular set of analyses or whatever. And lastly, does it have wide acceptance in the scientific community? If it doesn't have wide acceptance, this does not exclude it from court, but the judge may make a point of telling the jury, we are accepting this, but it has not yet been widely accepted by the scientific community. In other words, Dawbert is very similar to Frye, but it's a little bit more liberal in allowing new techniques, these sort of things, to be admitted to the court, provided the trial judge decides that that is okay. And I'll just point out that the use of DNA profiling in court as evidence in court would have been held up for many years under the Frye standard, because it would have been a very long time before every detail had been accepted by the wider scientific community. But under the Dawbert standard, it very rapidly became accepted as a method of analysis. So just to emphasize the point, please read this in your text. And this is the Dr. Coppolino's deadly house calls. This is a case where a brand new scientific technique was used in court and actually resulted in a conviction. Under the Frye standard, it would never have been admitted. They would have to go through all sorts of lengthy additional testing and analysis and publication and peer review and everything else. But here the court decided that their work was good enough to be accepted as a technique, and thereafter this became more widely used as an investigative technique. We talked a lot about all of the other kinds of the ancillary experts and everything in the lab. There are just a couple more to mention. And these are the fields of forensic psychiatry, which we hear of frequently because people often attribute the commission of crime to mental illness or whatever. And it's the role of the forensic psychiatrist to determine how the court should regard the person's claim, for instance, that they had no responsibility for a crime because they didn't know right from wrong or something like that. Forensic odontology. Now, forensic odontology involves using teeth to provide information. Very, very useful in cases of determining identity. If the person had a dentist who has dental records, then they are very frequently used to identify a body that is otherwise unidentifiable. One of the reasons being that your teeth are extremely strong and last much longer than the rest of the body. Even if a body is burned, for example, very frequently teeth remain, which can be identified from the dental records. Forensic odontology, however, also investigates bite marks. Bite marks are very frequently left in commission of a violent crime, and also, of course, in the case of sex crimes. But investigation of bite marks has come under a very dark cloud because it has been shown that it is too subjective. When it was actually scientifically examined, it was found that the error rate is too great, and that by and large, bite marks are not regarded as good evidence anymore. Forensic engineering, for us at the moment, a huge thing because of the collapse of the condo block in Florida. This is going to be extensively investigated by forensic engineers attempting to determine why that collapse occurred and whether there is liability for that collapse. Forensic computer science, we'll talk a little bit more about later on, and we'll also talk about forensic cell phone tracking. Here's something which I think we all know, but we don't really think about. As long as your phone is on, it is tracking your movement, and it provides a very good record. We can extract a very good record, in actual fact, of where you have been if you have had a phone with you which was on, switched on. So this has become a major area of forensic investigation. So here's another thing for you to think about if you continue to be interested in forensic science. That is that the simple gathering of evidence from a crime scene has become a very complex matter. In the old days, well, they would examine as best they could, try to determine what had happened by eye, do a report. They would gather maybe fingerprints or whatever. I just think of the early part of the 20th century. There was no point in gathering necessarily, even in gathering blood or whatever, because you didn't do DNA analysis, but you gather fingerprints and have a look, maybe look at the gun, see if you could determine which gun was used, etc., etc. Nowadays, people are going into crime scenes and they are vacuuming the floor. They are examining for hairs, single hairs. They are examining every blood stain, every possible biological stain. Just the gathering of evidence now requires expertise. And that is becoming the province of evidence technicians. These are people who are trained lab staff who are trained to go and recognize what may have evidentiary value and collect it under controlled conditions and preserve it under controlled conditions. And I believe that still there is no like federal guidelines for training or anything. So different precincts or different crime labs will train their own evidence technicians, but this may be something that you are interested in as a career. Okay, so that's it for today. And then tomorrow we proceed to begin to look at the crime scene itself.